Yes, so thanks for the kind introduction, David, even if you did mention quite how long I've been at James Cook University. And I want to say one thing. David was very kind in his introduction to me, but really perhaps didn't state emphatically enough Sylvan's contribution. Sylvan's really, none of this would have been possible without Sylvan's input. He's made enormous contributions to not only the Coral Genome project, but also to the assembly algorithms that will be more widely used for genome assembly. So we've been very fortunate to have Sylvan work with us. I've been very privileged to have Sylvan and a number of other people work with us for the last few years. So it's a sad day when he's moving on. So at the risk of this becoming a bit of a Genomics 101 lecture, I thought since we were bandying around these words, I should perhaps introduce some definitions. Genomics, when we're talking about genomics, we're talking about the study of all of the DNA in an organism, which includes all the boring bits, the stuff that doesn't appear in the protein repertoire. It doesn't appear in the obvious world of proteins that we live in. Whereas transcriptomics is the study of all of the transcripts, which are the more interesting parts of the genome, the genes which are doing things. So transcripts and transcriptomics, this is the study of the functional part of the genome. Alright, so for this audience, I don't need to convince you that coral reefs are spectacular places, spectacular and beautiful places, but of course things are going wrong. We've heard a lot about how things go wrong over the last few days. Reefs worldwide are in decline for a number of reasons, including diseases. So coral disease is a problem worldwide. It's not so much of a problem yet in Australia, I don't think, but it's been absolutely catastrophic in some parts of the world, and has been largely studied in terms of its distribution, its phenotypic effects and so on. We know very little about mechanisms of disease. And on the converse side, we know very little about how corals respond normally to the immune challenges which they must face day to day on a coral reef. We've heard that there are now very recently available coral genome sequences. There is a Japanese whole genome sequence for Acropora digitifera, as well as our Acropora millepora genome sequence, which Sylvan assembled. There are other whole genome sequences coming too. We can expect to see several more in the next few years. And indeed also for our coral, for Acropora millepora, Sylvan has assembled a transcriptome, a complete transcriptome, as complete a set of genes, transcripts, as is possible with a very large amount of next-generation RNA sequencing data. And this, as along with the genome sequences, we've been trying to apply to a number of kind of basic aspects of coral biology. So what do these allow us to do? What do these marvelous resources allow us to do for the first time or better? A number of things. First, they allow us to get some general characteristics of the coral gene set. In the last few years, it's become clear that the coral gene set is remarkably human-like in terms of the number of genes and also the kinds of genes that are there. This is one of the big surprises in evolutionary biology in the last few years, has been just how complex the genomes, the gene sets of very simple, morphologically simple animals are. So they allow us to count genes. They allow us to catalogue various known gene families by similarity to, for example, genes in the human genome. We can pick out many coral genes and get hints at what those genes might be doing because of their known roles in some other organisms. And we can do comparisons between these gene categories with other animals, and particularly informative for us is the comparison with the sea anemone Nematostella, which is morphologically very similar to corals, except that it has a handful of very different lifestyle traits. And that are affected by climate change or any particular challenge. We're now able to look at what happens to every gene in the coral genome. Many genes whose function we have no handle on the function of many genes because they have no similarity to genes in anything else. It doesn't matter. We can still see if those unknown genes are affected by any kind of challenge you care to name. So, the availability of coral genomes and transcriptomes, this is not just an academic exercise. It's actually got big practical benefits for beginning to tackle comprehensively how coral biology works. And I mentioned this comparison with Nematostella, with the sea anemone Nematostella. For us it's quite an informative, we hope it's going to be quite an informative comparison because of these big differences in lifestyle traits, the absence of a skeleton and the non-symbiotic nature of Nematostella. Perhaps allow us to infer something from the gene sets that are available in these two organisms. We can perhaps begin to get hints about what are the genetic underpinnings of these complex biological traits. And when you do this, Sylvain's introduced this idea already, one of the things you see if you compare the gene sets of Nematostella and Acropora is that there seems to be in Acropora a massive expansion of the sets of genes which are likely to function in immunity, in innate immunity. Sylvain gave the example of the NACT domain containing proteins. They're the primary intracellular pattern recognition molecules in higher animals. The toll-like receptors are the primary extracellular pattern receptors in higher animals. And whereas Nematostella has one canonical toll-like receptor and a handful of related receptors, Acropora has at least 4 of these toll-like receptors and many more of these interleukin-1 type receptors and orphan receptors. The same kind of thing holds for pretty much every domain that's associated with innate immunity in higher animals, so there are many more NACT domains, as Sylvain told you. I hope the numbers are the same as the ones Sylvain had. But also these genes, these domains which are implicated in apoptosis and other aspects of immunity are also overrepresented in coral relative to sea anemones. Which, one of our hypotheses is that when you're a colonial and symbiotic organism living on a reef, your life, it becomes much more important to accurately distinguish friend from foe. Okay, so back to coral disease and coral immunity. We want to understand, we've begun to tackle, using these amazing new resources, these molecular resources that we have, we've begun to tackle aspects of coral immunity and coral disease, hopefully. And we're coming at this from two ends. Really, we're interested in this process in terms of perhaps having implications for coral protection and management, if we can understand how coral disease is spread, what the vectors are, and so on. We're in a better position to do something about it. But also, we're also interested in understanding coral immunity from the point of view of trying to figure out aspects of how immune systems have evolved in higher animals. So our attack is two-pronged. Many people think that we have no interest in this. It's not true. We are actually interested in practical applications. But yes, it is true that we have an underlying interest in the basic biology of immune system evolution. And as part of this process, I was very fortunate to have a PhD student who's writing up at the moment, Yvonne Weiss, who came with a very strong immunity background. And she had the idea of comparing the immune response of coral with that of vertebrates to very specific immunogens. So there are a number of compounds in bacterial cell walls and so on which produce well-defined responses in higher animals. And the idea that we had, or she had, was to compare the responses, the molecular responses of the whole gene set in Acropora to that which we know occurs during challenge with the same kinds of compounds in vertebrates and flies. She did this on Heron Island, O's marvellous research station on Heron Island, a couple of years back, and the experimental protocol is relatively simple. What all she did was to simply introduce very small amounts of these compounds, these well-defined, chemically pure compounds, into the axial canals of a number of Acropora branches. Very dilute solutions. And then after a period of time, what we've done is to, so the treatments with these various compounds are shown here. So, of course, there's a couple of controls. One is the no treatment control, but we've also done controls where the coral's injected with sterile seawater to allow for the fact that actually the injection process causes some stress to the corals. In addition, we use 3 bacterial cell wall components, lipopolysaccharide, muramidol dipeptide, and tridap. These are bacterial cell wall components, and 1 viral mimic. So poly-IC is a mimic for viral nucleic acids. And because what happens next, so the biological experiment is cheap and relatively simple to do, but the follow-up is quite expensive. So we've spent some time working through these samples, and what I'm going to talk really about today is just one of the first step along the way of analysing these samples. So we looked initially at the response of coral to 1 bacterial cell wall component, muramidol dipeptide, and the viral mimic. And we looked in the first hour at what was happening, because one aspect of immunity that's particularly interesting is the short-term immune response, what happens immediately following immune challenge. And these, as I said before, these compounds produce well-defined responses in vertebrates with MDP signaling through NOD2, one of these NACH domain proteins, and poly-IC signaling through the toll-like receptor 3 in vertebrates. So this is just more or less what I just told you. We'll skip through that. And here's the treatment again. Take coral, inject it with compound, extract RNA, and then you sequence the hell out of it, basically. After an hour, break off the tip, extract RNA, and sequence using the next generation sequencer, the Illumina sequencer, 1 million reads, and simply count against the reference transcriptome or the reference genome what those reads correspond to as a sort of digital readout for gene expression. So this is an alternative to doing microarray type analysis. It's much cleaner kind of analysis than microarray analysis. You get a nice digital response. Counting reads seems to be a much more accurate and noise-free. I'm looking at Silvan. He's not quite so sure it's noise-free. However, it's a digital readout of gene expression. And the beauty is that we can look at the response of every gene. So here's what happens at the end of the day. With this experiment, there are only a limited number of genes that respond to the bacterial cell wall component and the viral mimic. One thing to notice is that the sets of genes that are responding are nearly mutually exclusive. The gene set that responds to the bacterial component is quite different to most of the genes that respond to the poly-IC treatment and viral mimic. And if you look at what genes are upregulated in this first hour of immune challenge with the bacterial cell wall component, it makes a lot of sense in terms of what we know about vertebrate immunity. Many of what I would call the usual suspects are upregulated. Mannose binding lectin, RAS GTPase, some tumor necrosis factor receptors. But intriguingly, these genes here, these proteins here, which are called GMAPs by some, IANS by others. So these are nucleotide binding proteins which are associated with immunity in vertebrates, whose roles in immunity really aren't too clear, but which are missing from most other animals. They belong to this big family of proteins called the nucleotide binding proteins, but they're quite distinct from them. They're distinguished from them by some unique characteristics, which are really in the subdomain structure of this characteristic domain. So this domain structure pretty much says that these coral proteins really are GMAPs. So 3 of the top 10 upregulated genes in corals are in this gene family, which is important in immunity in mammals, but which is missing from most other animals. What Yvonne did next was to confirm this result using quantitative PCR and to confirm that you get the same kind of response irrespective of which bacterial cell wall component you use. Remember, we used MDP, muramidol dipeptide, for the high throughput sequencing. You get the same kind of response of these genes, in fact stronger with lipopolysaccharide, and it's consistent across each of these bacterial cell wall components, whereas the viral mimic does nothing, very little, to expression of these GMAPs. So it's a response of the kind that you see in vertebrates to the same kinds of immunogens that produce that response in vertebrates. And this has really caught our imaginations because these proteins are very poorly characterized. They have a very scattered distribution across evolution. And a subset of the 8 genes that are present in man have clear functions in immunity. So immune functions seem to be shared between coral and man, which suggests that the role for these genes in innate immunity is very old. It dates back to the Cambrian explosion at least 540 million years ago. But the genes are missing from most other higher animals. They're missing from Drosophila, Caenorhabditis, and many other animals. Intriguingly, they turn out to be present, these genes turn out to be present in higher plants, and intriguingly, some of them have been implicated in immunity in plants. So this may be a family of genes whose roles aren't clear in vertebrates, but which have a role in immunity, but shared not only with lower animals, but also with another kingdom. So these may be the first examples of proteins with roles in immunity that are shared across this plant-animal divide. As far as I'm aware, there are no other proteins whose function in immunity is conserved. But there's another possibility, which is that we've got convergence on our hands, that evolution has been constrained in how it assembles proteins for this kind of role, and that it solved the same problem twice and come up with the same answer. So it's possible that this is convergence, but the complex subdomain structure of these proteins suggests that it may not be. And that's really where I want to leave it at this stage. As I said earlier, I've been very privileged to work with a very smart bunch of people over the last few years, and I'm just the mouthpiece for many people that are brighter than me. This work was done, the practical work was done by Yvonne. It was her idea to undertake this, but Sylvan has been absolutely crucial to the analyses and all of the bioinformatics. And I need also to mention my ANU collaborators, Eldon and David, who are involved in pretty much everything that we do with corals these days. Okay, thank you.